So this summer I worked with dipeptides, which are a class of small molecule consisting of two amino acids joined by a peptide bond. And in animals we know a lot about their function. It's been widely studied that they can affect everything from the immune and nervous system to antioxidants within the body. But in plants their study is relatively new, and we don't know a lot other than the fact that they can help regulate stress responses, and they could possibly alter the growth of the plant. So the goal of all of this research is to just generally increase knowledge about dipeptides, and we are doing that through inducibly overexpressing dipeptides like pepD, pepE, pepQ, and DCP. So dipeptides like pepD decrease dipeptide numbers because they take them and then they cleave them into their two original amino acids, whereas DCP does the opposite. It increases dipeptide levels by taking a longer amino acid chain and then breaking that up into dipeptides. And the very beginning of all this research is to transform bacteria with said dipeptides, and then you take those transformed bacteria to transform Arabidopsis seeds. And this is where I come in. So I started by sowing some already transformed seeds onto hygromycin antibacterial plates. And as you can see here, the mutated plants have a clear phenotype. You can clearly see that they are growing very well and almost normally on the hygromycin plates, whereas normal or non-mutated seeds are just dying almost immediately. And then we also had a bunch of already grown plants that I was able to genotype to confirm whether or not they actually had the gene insert. So here is the gel result from that. We had about 9 pepD plants, and out of those, roughly 5 contained the insert, and out of 4 DCP plants, roughly 3 contained the insert. So those plants, now that we know which ones have the overexpression, in the future we will be able to induce that to see what effect, if any, the altered dipeptide levels have on the plants. And there were also many more plants that were transplanted into soil from the hygromycin plates, but they are not yet grown enough to be able to genotype them. So in the future we can genotype those plants as well, and then induce the overexpression in them. And this research could have wider implications in the world of agriculture, for example, because if we can create plants that have increased stress tolerance, then we can create higher crop yields, and that could bring a lot more food to people. Probably just the environment. I loved all of the people there, and it was a very great experience as my first work in a lab. And I also got to conduct my own research and have my own schedule, and it was a really good insight into what I want to do in the future.